right, welcome. I'm Dr. Robert Wynn. I'm the Dean of the College of Arts and Sciences here at NMU. And it's my pleasure to welcome you here tonight to the Your Health Lecture Series. I had the good fortune many years ago to be in the initial planning meeting when leadership from Michigan State University College of Human Medicine and Northern Michigan University met to discuss the possibility of collaborating on a speaker series that would feature physicians and researchers from MSU who would come and speak here in the Upper Peninsula and include NMU faculty, staff, students, physicians and staff from the hospital, and members of the Marquette and surrounding communities. 15 years ago, October 1st, 2009, Dr. Jack Lipton discussed Parkinson's disease, origins, consequences, and potential therapeutics here on the NMU campus. The series was a big hit and is grown now to not only include NMU, but to include Michigan Tech, Bay College, and Lake Superior State University, which expands opportunities like the one we're having tonight to communities around the Upper Peninsula. I would like to thank the sponsors of our event tonight, Northern Michigan University, Michigan State College of Human Medicine, UP Health Systems Marquette. They've been involved right from the very beginning. And so with that, I'm going to turn the podium over to Mark Brevy, who is going to introduce our speaker, Dr. Hong. Mark.
Thank you, Dr. Wynn. And yeah, the Your Health Lecture Series predates my starting at MSU 12 years ago, and beyond even the UP now, we bring it to some of our early assurance program partners downstate. So you all have a lot to be proud of being the first and kind of leading the way. So again, my name is Mark Brevy. I'm Director of Community and Government Relations at Michigan State University College of Human Medicine. And we do really appreciate the collaboration and the 15 years of partnership. And really want to thank Dr. Wynn for the continued partnership. I would like to know at the conclusion of Dr. Hong's presentation, he will answer questions from the audience, both here in person. I'll bring around the mic to the room. And then also, if you're joining virtually, I will keep an eye on the Q&A as well. With that, I'd like to introduce our speaker this evening, Dr. Chaz Hong. And I learned earlier that once you meet Dr. Hong, he wants to be called Chaz. So you've all met Dr. Hong now. When you see him again in the future, make sure you call him Chaz. He is the chair of the Department of Medicine at MSU College of Human Medicine. He is honored to lead the MSU College of Human Medicine, Department of Medicine at this transformative period with exciting opportunities in discovery, innovation, education, and patient care. His vision is to help make MSU a dynamic innovator in academic medicine, attracting and developing future leaders who will transform the innovation landscape across Michigan and build a more just and healthy society. Dr. Hong is a Detroit native. He was here in Marquette about a year ago. Earlier today, he made his first trip to Sault Ste. Marie. So even though he's a native Michigander and native Detroiter, I think this is a couple of the first times that he's been in the UP. So we brought him during a good time of year to enjoy it. Completed his bachelor's degree at MIT, his MD, PhD in internal medicine residency at a pretty good school, Yale School of Medicine, his cardiology fellowship at Massachusetts General Hospital, and his research fellowship in chemical biology at another pretty good school, Harvard Medical School. Dr. Hong joins MSU from the University of Maryland School of Medicine, where he served as the Melvin Shiraki Professor of Medicine and Co-Chief of Cardiovascular Medicine. Prior to that, he was on faculty at Harvard Medical School and Vanderbilt University School of Medicine. And most importantly, Chaz is a lifelong Spartan fan. Dr. Hong.
Is the mic on? Okay, great. Thank you. Boy, if somebody could actually take a picture of me, I do post a lot of Twitter and things like that. We'll have some fun, okay? it really is an honor to be here. I think I was joking, but I'm pretty serious. I think I grew up in Detroit, but I'd never left 20-mile radius of the city. And one of the things that I'm really learning is that Michigan is a beautiful state, but it's also a big state. I mean, I had no idea that Sault Ste. Marie and Marquette was 3 hours away, but it's beautiful. I saw the locks for the first time. I went along the beach, and the sands of the beaches here. It took a little bit longer than 3 hours because I stopped along the way. It's actually more beautiful than the ones you see in California. So it's really beautiful, and so I really am enjoying, and I want you to ask questions, okay? And I think that the big thing for me is that my career has always been ups and downs, but it's always driven by curiosity. And if you guys are wanting to be a doctor, scientist, or a nurse, or any kind of health field, it's actually very important that you stay curious. Because if you're not curious, you become judgmental. Guess where I learned that from? I learned that from, I stole it from Ted Lasso. If you remember Ted Lasso, that show, be curious, not judgmental. And I take that to heart. And I think that's my secret sauce. I was always curious. And if a doctor is not curious, then that doctor would become judgmental. Instead of saying, why are you smoking? Right? What makes you smoke? What makes you do drugs? That kind of stuff. What's going on? Right? If you don't ask that question, you become judgmental. Oh, you're just stupid, right? And when you become a doctor's a judgmental, that breaks the therapeutic bond. So remember, and also if you're curious in medicine, there's so many things we don't know. And so you'll have a very wonderful time chasing after things that you don't know, right? And it's a wonderful field. That's why I'm a scientist, but I'm also a doctor. And it's a perfect fit for me because at the end of the day, I'm curious, okay? And I hope that you remember that as you journey on in your careers. I do have, as a physician, I have to make some disclosures, and it's a lot of fun. I have a lot of patents, and I have a startup company based on what I do. And it's going to be two outlines. I'm going to overview of cancer from an undergrad perspective and a patient perspective. Those are important. And then I'm going to talk about my science, which is sort of a novel death pathway, ferroptosis, and also acid-sensitive signaling as a pro-survival oncogenic pathway. That's important to me now, but I'm hoping that it will one day be important to everybody. In science, you just don't know, right? So it's gonna be in two talks, two parts. So I know this guy. I know this is being recorded, so I can't say a lot of stuff. Sid Mukherjee was my resident, so I know this guy personally. But I think he was writing this book when he was resident because he was a little distracted, to be honest. But number two, cancer is a number 2 leading cause of death. Okay, but number 1 is heart disease. I'm a cardiologist, so I could always say we're number 1. We're number 1. Cancer is number 2, but it's not really that clear because it is number 1 cause of death under 85. So this is number 1 cause of death for people in this room. Okay. But many cancers, if you take anything away from this lecture, is this. Many cancers are preventable. It's not just bad luck. It's preventable. So this is a lung cancer mortality, and something happened in the 80s where it started dropping like crazy. Lung cancers are not less common than it used to be. So what happened, right? Exactly. But people just didn't stop smoking. They didn't want to stop smoking. I remember when these were all controversial. Controversial as in the 60s, the cigarette company said there's no evidence. The evidence is weak. You need to do more research, right? They were kind of dragging their feet. We hear that now with climate change. We're not sure yet. Like one more. And they dragged this thing for about 22 decades, right? finally, Surgeon General came in and said, hey, the evidence is irrefutable. It causes cancer. And then I remember when I was in college, there was a smoking session in planes, and that was at the back of the plane, right? And if you were unfortunate and ended up in the back of the plane, it was very smoky, right? And there was a concept of, no more non-smoking. And these public health measures that was rooted in evidence, scientific evidence, really made the lung cancer drop, right? Now, there's vape. I just found out that my own kids do vape. I was like, what? We don't know what vape's going to do. It's probably better than smoking, to be honest, but probably not. In 10, 20 years, we might find that it does something to you. We just don't know. So there's a lot of things that are happening, but bottom line is that cancers are preventable by public health measures and by evidence-based public health measures, right? This is, there was irrefutable evidence. And then what happened here? This is a cervical cancer. And the cervical cancer is kind of interesting. Anything that happened before 1920s, who knows? I mean, people are learning to do this, right? So the evidence is kind of sketchy. But cervical cancer at one time was #1 cause of cancer death in women. Cervical cancer was. So if you remember the HeLa cell, that's the first human cell line that we learned how to grow. She died of cervical cancer. That's a cervical cancer. So what exactly happened here? What is this, right? Now, what happened during the 20s that really made a difference? It turns out that a doctor invented this thing called Pap smear. Pap smear was doing a cervical kind of smear and looking for precancerous lesions in women. And that came in 1928. And they were able to detect cervical cancers earlier and earlier. And that's why the rate plummeted in 1988. But it's actually further down. It's actually even lower now. Guess why? We've since discovered that cervical cancer is caused by human papillomavirus, subtype 16 and 18. It causes cervical cancers. And so now we have vaccines for that came out in 2006. This is something that my daughters, my children don't have. They're older than you. It wasn't around. But now I think it's a part of a childhood immunizations so that the cervical cancer would be eradicated in our lifetime. Now, nobody gets Pap smears anymore because we know what causes cervical cancer. It's caused by a virus, so you get a detection for virus. And if you don't have virus, then you're at high risk. They might give you a Pap smear. But if you're negative for HPV, you're safe. It's that almost deterministic. It's pretty amazing what's happened in the past 100 years. And then colorectal cancer is the most preventable cancers that we know. The diagnosis is about 150,000 cases a year. It's like 2 Michigan State stadiums full each year. The new diagnosis of colon cancer. But if you catch it at stage 1 and stage 2, it's a 90% survival. Basically, it's cure. It's not a lethal disease. Okay, 5-year survival, 90%. That's pretty darn good odds. But it's also the least prevented disease, cancer we know, because people are still dying at 50,000 deaths. Because if you catch it late and stage 4, game's over. It's a 90% mortality. So, what's going on? It's because people should be getting colonoscopy, screening colonoscopy, but they're not getting it. Either there's not enough doctors, or people just don't want to get it done. It's kind of, it's kind of uncomfortable, right? So in a rural communities, for instance, I think the Cologuard would be something like this would be very important. Where how many of you seen the advertisement Cologuard? Yeah, right. There's a SNL skit on Cologuard as well. And I think this is going to be transformative. Things like this will come up the pipeline where we will be able to do molecular diagnostics and find cancers before they become cancerous, right? And so there's a lot of exciting things happening. And then there's been treatment breakthroughs. Who knows who this Lance Armstrong is? I guess I should have just hit it. And then, Lance Armstrong, who knows, who remembers Lance Armstrong? Good, right? In 1996, he was diagnosed with stage 4 testicular cancer. He had a mets to his brain, right? But he was treated with cisplatin, the most successful chemotherapeutic agent of all time, and it was cured, right? So as far as I'm concerned, I don't care that he won 7 Tour de France's. I don't care that he cheated. But the fact that he's alive is a miracle, right? He's alive because of these scientists from Michigan State. Rosenberg, Van Camp, and Dimitrov Rosenberg and Van Camp actually discovered cisplatin and carboplatin, the 2 most successful chemotherapeutic agent of all time, bar none. And so that happened at Michigan State. And so there are a lot of exciting things that are happening in the pipeline. And this is actually like eye-popping. In 1974, if you have testicular cancer, you're a dead man. 95% dead in 5 years. Now, nobody dies of testicular cancer. So it's kind of remarkable how much things have changed through medical breakthroughs in medical science. So I want to let you know, just look around. There are a lot of diseases that are out there, ALS, Alzheimer's, heart failure. There are a lot of diseases that we don't have a cure for yet. But you will, in your lifetime, there will be a cure. And you will, I hope that some of you will play a part in that discovery and transformation. I've seen that many times. I've seen HIV. If you had an AIDS, you're a dead man, dead woman. No longer. It's managed because we have drugs to manage those things. So I'm hoping that in the future, even all cancers will be managed and find a cure for. But not all cancers at this time can be prevented or treated. This is a pancreatic ductal adenocarcinoma, PDAC. But for my purposes, I'm going to call it pancreatic cancer because this is like a vast majority of pancreatic cancers. And it's a second leading cause of cancer death in the United States. I forgot what the number 1 was. It's number 2. It's number 2. So it's kind of up there. And usually it's diagnosed very late because the pancreas sits actually in the back of your kind of retroperitoneum, deep inside your gut. So unless it's advanced, you don't have symptoms. And symptoms could be like symptoms of jaundice, abdominal pain. And by the time you have symptoms, the cancer has already spread. It's too advanced. And only 15% are eligible for curative surgical resection. 85% is already too late by the time it's diagnosed. And because of that, there has not been an improvement in survival for 30 years. So it remains flat at 13%. Okay. So these are the people that died of pancreatic cancer. Basically, if you get it, it's a death sentence. I think some of these people you probably don't know, but Jeopardy, Ellis Trebek, right? And Ruth Bader Ginsburg, and that Rickman from the Harry Potter series, right? So these are real people that who die of pancreatic cancer. So, I, as a chair, have been aggressively recruiting scientists who can help us cure cancers. And so we have the pancreatic cancer turns out to be driven by 90% of the pancreatic cancers are driven by activating mutations in this thing called RAS, right? And that is, RAS is normally important for cell growth, but when it's aberrantly activated, where it's activated too much or all the time, you end up with cancer. And so there's been a lot of therapies to prevent this RAS activation. But I have to brag. We recruited Dr. Jenny Klomp and Jeff Klomp from University of North Carolina. They're actually graduates of a Grand Valley State University. And they came back as a conquering heroes to our Grand Rapids campus. And they have science papers. In science, Science Magazine is like the top journal in our field. So you have a Jennifer Klomp and a Jeff Klomp as the first authors. I don't have a single science paper. So these are like super rock stars. So it's like really fortunate that we recruited them. And what they've shown is that the key target is ERK. And so the idea is how are we going to target ERK and how are we going to treat cancers by ERK? Everything centers on ERK. There's like a really groundbreaking work that even the NIH director noticed in her blog. offers hope for continued treatment advances. So it's really amazing, and hopefully they will follow up with novel therapies that could actually make impact on patients with who are diagnosed with pancreatic cancer. And research is this: their work is not going to cure people with pancreatic cancer today. Those are dead people, but they will cure people who will get pancreatic cancer in the future, right? That's the power of research. So are there other Achilles heel of cancer, right? Cancer cells are unique because they're like Frankenstein of cells. They're energetically very active and energetically very bizarre. So they're like nuclear submarine where you have a ton of power. Nuclear submarine can stay in underwater for years. They only come up so that the sailors could eat something, right? They could stay there forever because the submarine nuclear power is so powerful. But you could also, the nuclear powers could be also dangerous. So cancer cells, you could think of it as a submarine, nuclear power submarine with a little bit of leakage. Something is constantly going wrong. It's not a perfect system because there's always some radiation or little minor things that are going on. So that's how you could think of it as a cancer. But despite all that, mostly oxidative damage, cancer cells are stressed, but they're somehow protected from dying, okay? And so is there a protective signal that prevents cancer cells from going haywire? Because that was a question that we were asking. And so this gets to a story of Otto Warburg. Otto Warburg was a German scientist, and exactly 100 years ago, he described this phenomenon. If you ground up the cancers, what he found is that in contrast to normal cells, where you get a glucose that undergoes glycolysis, makes pyruvate, and then pyruvate gets shunted over to mitochondria to make ATP by oxidative phosphorylation. And how normal cells, when you don't have oxygen, they go through glycolysis, so where you have pyruvate, but instead of going to the mitochondria, they stop, and instead they produce lactate, right? That's what happens if you get Charlie horse, right? If you run too much, you get these cramping in your legs. It's lactate buildup, right? But in cancer cells, even in the presence of oxygen, they undergo preferential aerobic glycolysis, so they generate lactate. He got a Nobel Prize for that, but we didn't really know what this was. It was just a description. And so, but clinically, Warburg effect is very important because the consequence of this is that cancer cells are very glucose hungry. They're just starving for glucose. And so if you look at cancer PET scans, those cancer tracers that we use is glucose. It's a radio-labeled glucose. And when you see a PET scan, the cancer cells don't glow. It's cancer cells are taking up glucose like crazy, because that's a consequence of Warburg effect. And so it turns out that glioblastoma cells are professional Warburg effect cancer cells. They take up glucose like nobody's business. And what is cancer cell? Glioblastoma multiforme. It's the most common cause of brain cancer and has a green prognosis. Less than 5% survive 5 years. And there's no curative therapy. And because it's in your brain, it changes your personality. It changes who you are. It's really kind of awful cancer to have. And even if you have a response initially, it comes back 100% of the time. And when it does come back, it's invariably resistant to any treatment. So it's a horrible cancer. And there was only 2 FDA-approved drugs so far for glioblastoma, temozolomide and VEGF inhibitor. But look at this, they improved their survival by only 2 months. 2 months to 4 months combined is probably 6 months. And these were approved when I was in medical school. That was a long time ago, right? So there's not been much advance in our field. And these are the people who had glioblastoma die recently. I'm the child of the 80s, so I remember Gary Carter in 1986, so I put him there. But John McCain just recently died of glioblastoma. And Joe Biden is the son of Joe Biden. So, if you have glioblastoma, it's usually a death sentence. Let's go back to Warburg effect. So you have aerobic glycolysis, even in the presence of oxygen. Instead of going to oxidative phosphorylation, it goes through glycolysis. So what's the consequence of that? You generate lactate, but the lactate gets pumped out to the extracellular space along with a proton. And so the tumor environment becomes acidic, about pH of 6.5. That's really not a pH of like stomach acid, but it's like lower than normal. Normal is about 7.4. And so it's been well described that advanced cancers have a pH of 6.4, 6.5. And it's thought that this acidic tumor microenvironment plays a key role in promoting cancer cells to grow, a therapeutic resistance, and cancer spread, and also tumor immune response. It hides the cancer cells from immune surveillance. Those are the body of literature that are out there. And the question is there a signal? Is there signal that connects Warburg effect to cancer cell survival? That is kind of like the official question I'm asked. The reality was that we already had a drug and we had a hammer and we're looking for, this is how science is done. We already had a hammer and we're looking for a nail. The hammer, I'm going to talk about it later, but hammer is that we had a receptor. We already had an idea, hypothesis that there's a receptor that's mediating this effect. I'm going to tell you that story a little later. So these are pH reporters that we flipped it on the outside. So it's detecting pH on the outside of the cell, not inside the cell. But if you actually grow these tumors, brain cancer cells, in these clusters, they form these tumor spheroids. And basically what this is telling you is that inside the tumor is acidic. It's glowing yellow because pH is lower. Then even in a neutral pH media, inside the tumor cells are at 6.5. So this is a Warburg effect happening in the Petri dish. And glioblastomas are well known to acidify media because their Warburg is like out of control. And that is exactly how this Warburg is happening. So it was really exciting for us. And so we had a hammer, I told you. It was we had an inhibitor of a receptor called OGR1. Now it's called GPR68, but it was originally called OGR1. And it senses, it's an inhibitor of a receptor called, it's a class of receptor called G protein coupled receptors. And G coupled receptors are evolutionarily conserved receptors, 7 transmembrane proteins that you find in bacteria also. Bacteria rhodopsin, that senses light, is a GPCR. All your smell is all the smell receptors are GPCR. Most of your taste receptors are GPCR. And so GPCRs are evolved to sense our environment. And one of the GPCRs happens to be a proton receptor, proton sensor. I didn't know this. Like until we discovered this compound, I thought, oh gee, cells kind of sense acid because it's acid. But it turns out that there's a taste for acid in a cell. So this, you could think of it as a taste receptor for protons. And it's an interesting receptor because at pH 7.5, it's completely inactive. Our body, our blood is 7.4. So it's inactive at normal temperature pH. But at 6.5, it becomes activated. That 6.5 is the tumor pH. So we thought that, this could be kind of connecting the dots here. This could be important for Warburg effect mediated cancer survival. Why does it call it ogomorphin, right? We call it ogomorphin because it's an OGR1 inhibitor. And when you're a scientist and you make stuff up, you have the right to name your compound. So we had fun. We called it ogomorphin. Who's going to argue, right? So I'm very proud of this name. Does it work? So if you make glioblastoma cells, put it in this, grow them, and then put them in a Matrigel plug. It's like just like a plug. Then the cancer cells start sprouting. It's just like what happens in brain. they start sprouting into the matrix. And if you put it in a lower pH, they sprout more. But if you use this drug to block that proton sensing receptor, then that sprouting is trimmed off. So we're very excited. But it's actually more. The size of these tumor spheroids gets larger when you put them on lower pH. Okay? It's just like what you would predict if Warburg was right. And you could actually slow their growth. It's not actually not slowing the growth. I'm going to talk about this. It's actually killing the cells and diminish that if you actually block that signaling with our compound. We call it OGM, but it's actually ogomorphin. It's like OGM for short. And so I remember when it started, everybody said, this is kind of stupid because you'll never find a cure for glioblastoma. And I think one of my mentors is a chairman of medicine at Johns Hopkins, Mike Weisfeld. And he simply said, when somebody's cocksure that this is one thing, that usually means that they're wrong, right? So he actually connected me to another person. He's a chairman of neurosurgery at Hopkins. And he said, I think you got some legs here. Let's just work with this. And so what we did is that this is actually temozolomide, is a drug chemotherapy that's it's the state-of-the-art drug for chemotherapy. And what we've shown is our compound is about 100-fold more potent than temozolomide. And then really exciting thing is that this drug is actually very brain penetrant. It actually goes to the brain across the brain barrier pretty well. And even in cancer cells that are resistant, like U138, resistant to temozolomide, the OGM can kill those cells as well. And the one on the right, what you show is that it actually sensitizes the cancer cells to temozolomide. So at a dose when temozolomide alone doesn't kill cancer cells, and at a dose when OGM alone doesn't kill cancer cells, if you put them together now, they can act synergy and kill cancer cells. So temozolomide as chemotherapy is toxic. So the idea here is that you could increase the therapeutic window for temozolomide so that people could take less of a temozolomide, have less side effects, and yet still shrink down the tumors. That's sort of the hypothesis that we have now. The really exciting thing is that so far, OGM kills every single glioblastoma cells we tested. It doesn't matter where it came from, Mayo, UPenn, Johns Hopkins, University of Maryland, and Henry Ford. It kills cancers. It kills cancer cells like nobody's business. And then those red arrows are the temozolomide resistant cells. So it kills all those cell lines as well. We're very excited about that. And then one of the interesting things about glioblastomas, that it's not just glioblastoma, it's a glioblastoma multiforme. It means that these brain cancer cells are shape shifting. It shifts shapes. It shapes who it is. Even within the same patient, some of them have like stem cell-like phenotype, some of them have neuronal phenotype, some of them have mesenchymal stems. They keep changing. That's why it's hard to kill these cancer cells. It turns out that when you do a principal component analysis using an RNA-seq, like one of those pathway analysis that I don't understand, but Dr. Barr and people that I work with understand, they can't even tell. Like if you did this principal analysis on 3 pancreatic cancer cell line, it will spit out these are pancreatic cancer cell lines. When you do this with glioblastoma cell lines, you can't tell because every line is so not different from each other. But what's really interesting is that the genes, how these cells respond to ogomorphin is exactly the same. And so the genes that are enriched are either unfolded protein response, ER stress, negative regulation of growth, and apoptosis. But another thing that showed up was ferroptosis. And this is where some amount of courage and like sticking out works. One of my master's students, I'm going to talk about her, she's going to show up in the picture later. She said, it has to be ferroptosis. And I said, I'm a cardiologist. I never heard of ferroptosis. Like, I don't want to do this. Like, move on. Like, I don't want to go there. I don't want to go into a rabbit hole. And the next week, she got a postdoc to say it is ferroptosis. So it was 2 versus 1, so they won. And so we decided, well, we're going to pursue this for 1 month. And so you have 1 month to show me that this is actually ferroptosis. That 1 month became 2 years. So this is fresh off the story. This is literally 2 years ago. I had never heard of ferroptosis. This is all new. The reason is that Jessica was a graduate student, and I only knew one thing, one way to kill cancer cells. It was apoptosis. I didn't know there was a ferroptosis. I didn't know. I'm a cardiologist. So when she said this data, I was like, well, positive control is weak. Well, you did this wrong, blah, blah, blah. I spent 6 months. I forced her to do 6 months to repeat the same experiments to show that it's apoptosis. And then finally she said, Chas, it's not apoptosis. Right? And then I said, why can it be? And she said that, ferroptosis. It's like, okay, these are the culprits. So can ferroptosis really be involved? What is evidence, right? Here's a professor saying it's not ferroptosis, never heard of it. And so how do you have to convince a skeptical boss that I'm wrong, right? So here's the evidence they provided. All the ferroptosis markers are going up. And then the ferroptosis suppressors are going down, right? Was it enough? As a professor, I said, no, this is not, that doesn't mean anything. Like genes going up and down, what does that mean? It's really not ferroptosis. Prove to me that it's a ferroptosis. Otherwise, I'm not going to pursue this angle. And so what is ferroptosis? I had no idea what it was. Basically, it is non-apoptotic, iron dependent. That's why it's called ferroptosis. Fer is in iron, and oxidative cell death. That's the definition. This is not necessarily the nature's definition. It's not necessarily the creator's definition. It's just our working functional definition. Is that it's not apoptosis, sort of requires iron, and it's oxidative stress. That's sort of working definition of ferroptosis. It's our definition, not nature's definition. So how do we know what is ferroptosis? What are the features of ferroptosis? It was actually discovered by Brent Cochran at Columbia University using Erastin. Erastin is one of those weird drugs that kills cells, but it didn't kill cells by apoptosis. So he thought, let's study this. And then when he discovered that the drug that he discovered turns out to be an inhibitor of this channel that is the cysteine uptake channel, like a pump exchanger. So why is cysteine so important? It turns out that cysteine, I'm giving you like a biochemistry 101 in like one lecture. This is like, if you know this, don't worry about it. This is not on your exam. This is a lot of material. But cysteine is the main ingredient for glutathione. And glutathione is our main antioxidant in our cells, okay? So if you block cysteine intake, then the glutathione goes away. And then when that happens, there's oxidation of lipids. And lipids that are oxidized are highly reactive, highly dangerous. It's like, and then you end up bursting mitochondria. That's sort of a very kind of cartoonish version of what ferroptosis is. But the important thing is that there are specific markers that we could tell whether ferroptosis is occurring in a cell. Glutathione levels go down, like oxidized lipid species goes up, and then mitochondria break down. So I asked Jessica, I don't care about whether genes going up and down. You have to show me these three things that are happening. And it turns out that, lo and behold, glutathione goes down in cancer cells treated with glutathione, ogomorphin. And then oxidized lipids go up. And I didn't know how to read this. It didn't seem very impressive, but the chi-square is about 1,000. Anything over 4 is considered significant. So this is like hitting it out of the park. So this is lipid peroxidation occurring in real time in cancer cells that are treated with ogomorphin. And then the mitochondria in cells that are treated with ogomorphin are blown up, just like what you might see in ferroptosis. So at this point, I said, okay, you win. It is ferroptosis. So what we've done, despite my best effort to stop this story, is that we've shown that GPR68 is an autocrine pro-oncogenic pathway acted by Warburg effect, okay? We just found it's a hypothesis. It has to be validated by army of scientists. We have hypothesis, and we have evidence that supports our hypothesis that the way the Warburg protects cancer cells is not cell autonomous. It's actually autocrine. There's a proton that gets pumped out and it activates the surface receptor, then that triggers cancer cell survival pathway. That's a big story that, a lot of it, we're skipping a lot of the data here. But if you block that pathway in cancer cells, now you have ferroptotic cell death. That's what we discovered. So we not only discovered a novel pathway for cancer cell survival, but we also found a pathway where you could kill cancer cells. So really killing two birds with one stone. I was very happy with it. And it's unique among ferroptosis inducers in a sense that it doesn't touch normal cells because, it only kills cancer cells that are activated by GPR68. And so we are very excited that this is how are we going to get cancer cells that are highly metabolically, altered and target these cancer cells. And this is our proof that our brain cells, normal cells, where it's not acidic, they actually don't care. You give this compound, and it's not chemotherapy. The receptor's not activated, so you can't inhibit something that's not activated. So they're protected. They're not dying. And so I'm going to skip this mechanism of cell specificity. I'm partnered with many people trying to figure out where does specificity come from, because the traditional ferroptosis inducers are poisons. They're just like chemotherapy. Anytime you get rid of glutathione, that's poison. Right? It's going to kill cells. So we don't want that. We want, how does this work? And this is where the scientist in me, as a curiosity-driven person, we have to figure this out. It's a fun pursuit. And this is an unexpected element that, if you remember, its analogies turns out to be apt. Submarine, it's like cancer cells are submarine with a leaky reactor. But somehow people have been patching it together and the cancer cells are kind of living and surviving and thriving and, beating out the normal cells. But there's something going wrong. And Tom O'Halloran is the director of the Institute for Elemental Medicine at Michigan State. And we partnered with him. And we found an element that may trigger this chain reaction. So we're very excited. Tom, being a better scientist, being a good scientist means you have to be critical. He's refused to believe this, so we have to prove it like 10 more times. So, but I've already, walks like a dog, quacks like a dog. I'm convinced, but I've yet to convince him to that this is actually real solid science. But I'm 100% sure I'm be able to, put him around. It just needs more data, more experiments, more repetition. But it's really exciting for us. We think that we found something that causes short circuit. And so the reason why I came here to Michigan State is because we have one of the best medicinal chemist groups, academic groups. And there's one group at Midland, Michigan, that are making compounds. We have an entire department of chemistry that are experts in drug discovery. And so the idea is that my job talk for Michigan State was cisplatin 2.0. So, it's a little bit of delusional, but if you want to do something great, you have to be a little delusional. You have to believe in your stuff, right? So I think I believed it, and I came here for that reason. And Michigan State also had a veterinary school. And dogs get brain cancer, glioblastoma, it turns out. And it's the same brain cancer. Our compound actually kills dog brain cancer cells too. So lo and behold. And we are a partner with cancer doctors at Henry Ford. And the idea is that we want to bring cancer therapy as quickly as we can to patients who will get cancer tomorrow. My inbox is full of people saying, my wife just got cancer a month ago. When is the clinical trial? It's too late for her, right? This won't happen. But this will cure cancer for people, brain cancer. I hope. The hope here is not real yet. Hope is that these compounds, or Me Too compounds or imitation compounds that come from drug companies, will be a cure for people who will get cancer in the future, brain cancer in the future. And this is to show that it also works for pancreatic cancers, because the Warburg effects is like revved up in pancreatic cancers as well. And the exciting thing here is that the radiation acts in synergistic manner with ogomorphin. So I think it's very exciting stuff. And there's a real world evidence as well. It turns out that lorazepam is anti-anxiety medication, actually activates GPR68 receptor. The proton receptor gets activated just by accidentally. So if you take alprazolam, which is another anxiolytic medicine, it doesn't activate GPCR. But people have actually looked at electron health records, and they showed that people who take lorazepam versus people who take alprazolam or nothing, they do worse when they get pancreatic cancer. So I think we have a real world evidence that this is not Chazong dreaming, but there's something we're barking up the right tree. And so there's other lung cancers because Warburg effect gets activated in most advanced cancers. So this is anytime you use PET scan to detect metastatic cancers, we think that this is going to be effective. And also immune cells. There's this is like a shoulder injury. There's a story of emerging story of inflammation as well, and that this is inflammation. So nothing in nature happens by accident, right? Like there's no GPR68 didn't evolve to kill us by cancer. There's a normal function for GPR68. So what is it? So we hypothesize that it protects, this exists to protect our organs from oxidative stress when that occurs. So what are these oxidative stresses like? It turns out that when you have stroke, our pH in our brain actually drops to 6.5. That's precisely the pH when this receptor is activated. And so there's actually a little body of literature now saying GPR68 is a neuroprotective signaling. So it's actually a good thing in the context of a stroke. It's probably a good thing in the context of heart attacks as well. And so we're working on a drug to protect brain during stroke as well by overactivating this receptor. So it's really exciting. These are the collaborators. It takes a village to make a story. And Jessica, even though she's happy, she obviously didn't like me enough to come with me, so she stayed. But we do have Chuck Williams and Leif actually came with me. So I think thank you for your attention, and hopefully you learned a few things and was excited about other aspects of it. And I'll take your questions.
All right, any questions in the audience? I have some online too that I can go to.
Great.
All right,
Yes,
we'll
sir.
go Dr. Wynn first.
First, thank you very much for a really, really interesting talk. So you already, excuse me, you already answered this part about, because I know that lots of cancers undergo the Warburg effect, but what does ogre-morphin do in normal tissues that do glycolysis? And I'll give you an example. Skeletal muscle. Skeletal muscle cells are happy to just use glycolysis, particularly when you're exercising at a high level. So what does ogre-morphin do to those cells?
That's a great question. We don't know. And in fact, when you put these pH sensors in our animals, these skeletal muscles light up. And it lights up in T tubules and in membranes like crazy. They just look like cancer cells, right? So the question is, if you were to inhibit GPR68, will they have muscle wasting disease? Will they have muscular dystrophy-like phenotypes? That's a great question. We don't know, but there are actually people. There are knockout mice in GPR. They lack not GPR68, and they're completely okay. And they're actually resistant to pancreatic cancers and ovarian cancers and melanomas. So it fits in our story. There are people walking around who have no GPR68 because in Middle Eastern, I think it's Turkey, there's a culture where you have first order cousin mating because there's remote parts of mountains of Turkey. And then they tend to have a lot of these rare mutations that actually, and there's people who are homozygous. And the crazy thing is that they have poor dentition. They have something wrong with their tooth enamel, but other than rotten teeth, they're actually perfectly fine. We don't know whether they have less tendency to get cancer or not, but as far as I know, muscle weaknesses have never been seen in mice, and muscle weakness have never been described in people who lack this gene. So we think it's okay, but we don't know. Thank you. Yeah, I mean, the beauty of science is that, like, you answer one question, there are two questions, one answer question and two answers question. So you could go to town forever. I love it. That's why I love science.
What are your thoughts about
VEGF, right? Yes. I think it's good. I mean, I think I wish that it worked better. But 4 months is not that great, right? But the truth of the matter is that people who are getting it think that they're going to be that 5% who will outlive the cancer. But I'm not sure whether patients who are actually getting these drugs know that it extends their life by 4 months only. Right? My father had cancer, and you're so desperate, you hear what you want to hear, right? So it's not a cure. Right.
Hi, my name is Grace. It's very nice to
Nice
meet
to meet
you. I had a question about the how would you ingest this drug? How would it be given to you? And would it be given to you coupled with the temozolomide, considering that does help, or some sort of iron therapy, considering that would be helping the pathway?
That's a great question. Okay, so to answer that question, we don't know yet. Right? We haven't paved that road yet, but that question is a key question, right? So one thing we're going to do with Henry Brem is that he's world's expert on putting these drugs in little sponges, dissolve it and then put it in. So when you take out the chunk of brain tissue, cancer brain tissue, he's going to implant that into the brain. So it's going to be locally delivered. And you got to realize that if you remember, it actually synergizes radiation therapy, synergizes with temozolomide. So the idea is that while people are getting those therapies, you may give them by mouth, or you may just implant it and then give them radiation and chemo. We don't know yet. We have some issues with orally bioavailable. That's about a $50 million question. So we don't have that answer yet, but that's how we imagine that this will work. And this may be similar to like chronic myelogenic leukemia that's caused by what they call Philadelphia chromosome. There's an ABL1 kinase, and it used to be universally fatal. But once they came up with an ABL1 kinase drug, people take that for life. So it's not like these cancer ever go away, but it's suppressed. So you live with cancer. So we think that it probably not be a cure. It could be a cure with a chemotherapy and radiation therapy, but I think that the second approach will be a suppressive therapy, where you have to take these drugs by mouth, hopefully, so that the cancer doesn't come back.
Having brain cancer, would Optune enhance or help the illness?
Pardon me?
Having brain cancer, would Optune enhance or help the illness?
I don't know what that is. So
Okay.
I don't know what that means. Sorry about that.
I'll go to the next one then.
Okay.
How does PIN1 or STAT3 or mTOR play in phoroptosis?
Boy, that's actually a good question. I know that they're playing in the same space. Ferroptosis can be induced by these mTOR inhibitors, but you got to give me a break. I only discovered this 2 years ago, so I can't answer at the top of my head. I've read it, but it didn't quite register. So excuse me.
Hi, I'm Sam.
Yes.
So, I was wondering how you got from cardiology to cancer.
Yeah, I get asked it a lot, and boy, accidentally, obviously, right? Literally, I think 2 years ago, 3 years ago, I was kind of upset because cardiologists, scientists are actually funded. I'm NIH funded to do something else. I was actually kind of upset that they're running into a rabbit hole that I didn't want to go into. But I'm smart enough to know when I'm wrong, and I'm smart enough to know when people are smarter than me, and I'm smart enough to know when it smells good. And so I just kept my mouth shut and followed what my students were doing. Now I take credit for what they did, right? I didn't go, like, literally 2 years ago, 3 years ago, I remember getting upset. I said, I'm not funded to do this cancer stuff. I'll never get a grant in Cancer Institute because I'm a cardiologist, right? science is like sort of like a click, right? So I didn't intend to do it. I didn't wake up and say, hey, I wanted to cure brain cancer. I just followed the lead of my students who are smarter than me. And if you remember that, you'll be fine. Just surround yourself people who are smarter than you and just learn to listen. And you know, and then later on, if you're the boss, you could take credit for their ideas.
What are your thoughts about Ben Williams from the 1990s beating glioblastoma and still living today? Does his off-label drug use keep him living?
Yeah, actually, there's always a outlier, and there's actually a lot of a group of people who had ketogenic diet and all that stuff, and they're like not a lot, they're like a handful of people that are alive there. And you know, I honestly, I don't know. I think it's misdiagnosis, or maybe it was a low grade glioma. I just don't know because he's still alive. We can't take his brain tissue, right? But there are outliers who get struck by lightning and still live. But there are a handful of people who lived 20 years of glioblastoma. And we would, you know, maybe they'll tell us something about what makes them special, but we don't know.
It seems like this one might be connected to what are your thoughts about Thomas Seyfried's approach with ketogenic and brain cancer?
Yeah, that's actually a great question. So there are people at Memorial Sloan Kettering that ketogenic diet was actually proposed first by Michigan State scientists, believe it or not, right? But if you look at the data, they're not everybody, but significant portion, like 30% of the people who were on ketogenic diet actually live. And there are people in that original study that are still alive years after that diet. The problem with ketogenic diet is that we don't really know how it works. We think they're the same pathway, because what ketogenic diet at the end of the day does, it deprives cancer cells of glucose, right? Ketogenic diet is just nothing but cream and like cream cheese, and it's just like disgusting thing, right? No sugar, right? So it makes sense. So in a way, we're barking up the right tree. It's just that ketogenic diet hasn't worked for everybody. We're not really sure. And it really hasn't taken like a foot, like really taken. I think it's just because ketogenic diet is kind of hard to stick with. I think that's the main problem. But there's evidence that it has potential. Right. Thank you. Yes. Well, I'm getting a lot of great questions. Thank you.
I know you said that the ogre-morphin is very cancer specific. Are there any other pathways that you have observed that aren't oncogenic that it has affected?
Boy, that's actually a very, very good question, because ogre-morphin, it hasn't been published yet, but I have a grant that it actually prevents inflammation associated with anytime there's a low pH. And one of them is ARDS, acute respiratory distress syndrome. And the reason why we got into, even though I'm a cardiologist, the reason why I got into this acute respiratory distress syndrome is that during COVID, unless you did COVID-related research, your lab was shut down. So we kind of said, well, you know, if you have ARDS, pneumonia, right? Acute respiratory distress syndrome, the lungs become acidic. It's not only hypoxic, it becomes acidic. And the pH is about 6.5. So we had a hammer. So it's like, well, let's test this. It turns out that when you actually block GPR68 in the context of, say, bacteria-induced ARDS in mice, we don't know in humans yet, it actually blunts the ARDS. It prevents inflammation. It prevents cell leakage. Endothelial cells are sturdier. So it seems to be, it may be involved in that regard as well. And other people have shown that GPR68 inhibition may be useful for like Crohn's disease. Anytime you have inflammation or focus, those areas are kind of hot, but they're not only hot, but they're also slightly acidic. So we think that's not my idea, but other people think that GPR68 is actually more involved in inflammation. Thank you.
All right, let's end with this one. Since Warburg effect occurs also in cells in shock, i.e. cardiogenic shock, do you foresee ogre-morphin having a role outside cancer space?
Yeah, I think that's a very good question. I think in the setting of shock, GPR68 is probably a protective mechanism. You probably don't want to block it. In that setting, you probably want to have an activator, right? So, when you could imagine if you're designing a clinical trial, you don't want somebody who has a stroke propensity. You want to make sure that, they don't have heart disease, things like that. You just want to control it. Because if you're on ogomorphin and have a cardiogenic shock, they'll probably kill you. I just don't know. But it could be good or bad, but my gut instinct is that it'll probably be bad in the context of shock. Thank you.
All right.
Wow, that's a lot of questions. So this is great. Thank you.
Let's thank Dr. Hong, or we should say Chaz,
Yeah, Chaz.
one more time. And want to thank again our hosts, Northern Michigan University, and especially Dr. Nguyen keeping this going for 15 years. And thank you all for joining us in person online, and have a great rest of your evening.
Thank you. So I guess some of the students I'll meet tomorrow.